We have a number of people on the Zoom already, and I'm sure there will be more that that join. I'm very pleased today to have two speakers, Doctors Hyder and Hansen. They will go in that order. And so first is Fahmeed Hyder, who is a professor of radiology and biomedical imaging and biomedical engineering. He received his PhD in biophysical chemistry from Yale, where he was also an associate research scientist and postdoctoral associate with Douglas Rothman. He's been in the faculty since 1999. Seems hard to believe. You look very young. And currently holds dual professor appointments in diagnostic radiology and biomedical engineering. Dr. Hyder is the director of the high field horizontal small bore systems at Yale's MRI Research Center. He uses magnetic resonance methods to map physiology and chemistry that underlie brain function for early disease detection, but also for targeted drug delivery and monitoring treatments. He's also the founder and director of Yale's Quantitative Neuroscience with Magnetic Resonance Core Center, the only NIH-supported programmatic effort at Yale on neuroimaging with MR technologies. His work has produced over 100 peer-reviewed publications, and he's written and edited books on functional brain imaging. I have to say something that I'm becoming increasingly interested as I age. He has received early career awards from various scientific societies and funding scientific agencies. For me, it's a pleasure to have you. Thanks for being here, and we look forward to hearing from you.
Thank you. Thank you very much for that generous introduction. I hope I can live up to that introduction. It's a pleasure to be here. Hopefully you can see the slide.
Yep, perfect.
So my topic today is a mouthful. Dysregulated transmembrane ion gradients highlight cancer invasion and proliferation. I believe the latter four words are probably more akin to a lot of the audience members, but I'm going to make the connection, or try to at least, for the initial part of this title, which is a mouthful. So a little bit of preface, I guess. I'm interested in brain metabolism. Why? Because they tell us about the pathways which neuronal and glial cells use nutrients to fuel their function, but also growth. I'm interested in how cells work, what their main nutrients are in different situations, different states, how they get these nutrients. That's actually a very important indicator of disease onset in a lot of cases, but also how they use these nutrients to generate energy, which is crucial for the brain, but also to regrow in disease, in damage, and so on. And all of these processes happen naturally for normal situations, but they also begin to malfunction. So these questions that I pose for myself, as well as my group members and my collaborators, these questions are fundamental for functional imaging of the brain, because the energy demand for normal brain work is extremely high. And that still is a very unique property of how this organ varies from a lot of other organs in the body. But these questions are also relevant in cancer, molecular imaging of brain disorders in general, but especially cancer, because this fundamentally is a disease of growth, and growth requires lots of fuel. And whenever you need to regulate fuel, compete for fuel, that is a metabolic question by itself. So that's my preface, but my main objective today is to talk to you about the importance of transmembrane gradients. They're crucially linked to cellular metabolism, and this property differs substantially between normal cells and cancer cells. Abnormal transmembrane proton and sodium ion gradients, specifically, I'll talk about proton and sodium ion gradients. They're consequences of pathophysiological alterations occurring at the cellular level. And the two primary things I'll focus on is acidity and salinity of the interstitial fluid. These two quantities reflect the use of proton and sodium ions respectively. They actually regulate essential cellular functions in health as well as in disease, especially cancer. And with regard to cancer, tumor evasiveness, which is one of the hallmarks, and even resistance to therapy, this is actually enhanced by acidic interstitial fluid, which is actually a consequence of upregulated glycolysis. I will talk about this in a few minutes. But there's also recent discoveries from our work and our work within related groups that show that enhanced proliferation, which is also a hallmark of cancer, is impacted by interstitial salinity. So both the acidity and the salinity of the interstitial fluid is crucial. A little bit of background. I think this background is probably redundant, but for the sake of being that, proton and sodium ions are vital for numerous processes, from maintaining blood pressure to neuronal firing. Homeostatically, we maintain a very large sodium gradient, almost 2 orders of magnitude, almost an order of magnitude, lending to a very strong transmembrane gradient for sodium, but similarly for proton gradients, which is usually measured in terms of pH, which, if you know, is just a logarithmic scale. So even though the pH between interstitial and intracellular compartments are 7.4 and 7.2, in pH units, it's a small value, but in terms of actual concentration, it's a very, very large gradient. There are various mechanisms which are located at the cell membrane help regulate the proton and the sodium levels to avoid these mishaps that the cells try very hard to avoid. First, mechanisms that regulate transmembrane proton gradients are shown here in a cartoon form. Let me just go around the cell here. First is this mechanism here, carbonic anhydrase 9 specifically, but these kinds of instruments are used to basically take out the carbon dioxide and water generated by aerobic oxidation and take them out, and it basically takes them out in terms of bicarbonate and protons, and which acidifies the extracellular space. Then there's these channels NBCs and NCBEs, which abbreviations are shown here. For the sake of time, I'll just go through the abbreviations. These bring in both bicarbonate and sodium, and this one brings in bicarbonate and takes out chloride ions. Both of these contribute to changing of the intracellular pH. MCTs, the monocarboxyl transporters, are of various kinds. MCT1 and MCT4 work in opposite ways. They bring in acids for MCT1 and take out acids for MCT4, altering hydrogen ion concentration in each of these compartments specifically. Important one, NET1, sodium proton exchanger, takes for every proton taken out, brings in the sodium, and that can alter the proton gradient as well. These are ATPase, ATP-dependent vacuolar proton ATPase, where you're bringing in, you're taking out protons to acidify the extracellular space, as well as these acid sensing ion channels, as well as these epithelial specific sodium channels. They also are known to affect proton, but as well as sodium, because a lot of these different channels have sodium gradients, and therefore these won't be repeated because these are the channels that I just talked about. But there are other mechanisms. This is a channel that I just showed in the previous slide, the NBC. The NHE as well is the sodium proton exchanger. All of these have a role in altering the sodium level in the extracellular space. This particular channel, the NCX, the sodium calcium exchanger, it exchanges sodium for calcium between the compartments. It's interesting, it was recently discovered, but in cancer specifically, this is actually mutated. In addition, there's this new channels mentioned here that are specific to sodium regulation across the compartment. The most important one of this is probably, and well known, is the sodium proton ATPase, which is exchanging of sodium and the potassium at the cost of ATP to basically maintain ionic balance to keep the resting potential the same so that the cell can essentially keep on doing what it's designed to do. So these are the mechanisms that are in play. The glucose metabolism is key to all of these processes. In the healthy neuropil, as blood brings in both glucose and oxygen in the form of oxyhemoglobin, glucose is its transport is expedited by these transporters, but oxygen goes through just passive diffusion into the cell. Just breakdown of glycose through the glycolytic steps, and then further breakdown in the Krebs cycle, generating large amounts of ATP. All of these processes generate small amounts of protons, but also carbon dioxide and water, which I have just mentioned. Carbonic anhydrase has a role in taking out these waste products. So this is complete oxidation of glucose, if you will, but even under normal conditions, there is some less oxidation than expected from theory, where some of the glucose is actually shunted towards biosynthetic processes, but some of the glucose is actually extruded in terms of lactate by the help of these MCTs that were mentioned earlier. A lot of the protons generated are removed, as I said, from these different mechanisms which are mentioned, NHEs, also carbonic anhydrases. But in cancer, in the unhealthy neuropil, a lot of dysfunction happens in terms of the amount of glycolysis versus the amount of oxidation that happens. And therefore, some of these aerobic glycolytic steps are augmented to the point where these machineries that I just mentioned with carbonic anhydrases, they're upregulated in MCT4s, they're upregulated to handle the extrusion of lactate more efficiently, but also in terms of the NHE, which is also upregulated to alter the proton in the extracellular space, the vacuolar proton ATPase, but along with some of the same mechanism, same machinery or additional machineries, you can also experience a lot of imbalances in the sodium environment. A key player, as I just mentioned, is the mutation of the NCX, which was recently discovered in cancer, but also the fact that there is actually downregulation of oxidation within cancer cells, the reduced role of potassium sodium ATPase is also an important role of causing these sodium imbalances as shown in this cartoon. So this is the unhealthy neuropil, and our role is to understand and observe the impact of the altered proton as well as transmembrane gradients. So tumors are highly glycolytic, even when well oxygenated. And this aerobic glycolysis phenotype enabled by metabolism of different nutrients, generates lots of acidic products that are extruded efficiently. For example, even carbon dioxide and water generated from oxidative metabolism they contribute to acidification of the interstitial space by these anionic exchanges like carbonic anhydrase. Interstitial acidosis actually helps with tumor cell invasion. They actually degrade the interstitial matrix, and they also promote angiogenesis while suppressing immune response. So this is an example from Bob Gillis's work where it is shown that whenever there's reduced acidification, this is done in a culture system where there's reduced acidification, there is enhanced invasion of cells in terms of tumor growth. This is an exemplary study shown in situ, but in vivo evidence for this from our lab and other labs have supported this observation. The impact of the altered transmembrane sodium gradient, normal cells maintain hyperpolarized membrane potential, but depolarized membrane potential in non-excitable cells is linked to their proliferation. I'll point to this in the next slide a little bit more. In fact, even immune cells can sense sodium in the interstitial milieu. Therefore, salinity of the interstitial and intracellular compartments may be crucial, as has been pointed out by recent work. It may be important for early diagnosis of cancer, but also maybe even tracking new cancer treatments. So this slide points this out. This is a scale of membrane potential. On the bottom are these non-proliferating cells. These are glial cells and neuronal cells. They have a very hyperpolarized membrane potential. It's the equation which takes into account sodium and the chloride ion concentrations. Up here are the proliferating cells, which are non-tumor cells, but the membrane potential measured of tumor cells of a variety of different kinds, you see they right up here in the depolarized level. So in essence, what a lot of observations from various groups have shown is that the weak transmembrane sodium gradient maintaining a depolarized membrane potential is actually necessary for these cells to survive in their altered environment that they create for their survival at the cost of actually normal cells that they're replacing. Whereas, these normal cells, especially in the brain, maintain strong transmembrane sodium gradients, and they may do so by maintaining a hyperpolarized membrane potential. So these are the hallmarks of cancer. Our goal, and my goal in the next few minutes, is to show that you can employ sodium and proton imaging methods to actually observe two properties, invasion and proliferation of cancer cells, by using proton and sodium imaging respectively. To talk a little bit about that, I'm sure this method is quite familiar with most. Oops. Just lost my slide. Sorry about that. Didn't like one slide.
Slide disappeared.
Yeah, I realized that. It just didn't like one slide.
Do you want to stop sharing and start again, or is that what you're doing?
Yeah, I'm trying to do that right now.
Okay.
Okay, so I'll skip that slide. Didn't like. And see if I can get from that. So the slide that I unfortunately had that, it bounced me out, a PowerPoint. Basically, this describes a chelate that is the design structure of most contrast agents that is used in clinically. The preferable design is where you take a gadolinium ion, it's very paramagnetic, and you use that to reduce its toxicity. You use a chelate with donating components like oxygen atoms that would provide the stability for conjugation, reducing the toxicity of the metal ion. But this chelating ion is some of the key of the types of imaging observations that I'm going to show you about. So this is essentially a proton spectrum of a chelate like this. This is it's called a DOTP molecule. It's basically a molecule that contains multiple phosphonate groups, much like phosphonates that exist in molecules like ATP and ADP. So this is a very common diamagnetic range of signals that you would see emanating for different protons, the non-exchangeable protons that exist in these carbons, as well as the carbons existing in these pendant arms. By the way, I point out that these phosphonates on this molecule like this, these protons exchange with protons of water, and that kind of exchange is actually a pH mediated, and therefore these molecules are essentially pH sensor, much like ATP an ADP is using phosphorus MR. So if you now chelate this, take this molecule and complex this with a paramagnetic metal, in this case, I'm not using gadolinium, but a thulium ion, you essentially cause this large expansion of the same chemical shift that you see here within a few parts per million, and that's expanded by hundreds of parts per million. So these signals have no potential of overlap with other types of signals that you may observe in vivo. And this is the water signal. And so this hyperfine shifted signal has very unusual properties. Basically, all it means is that you can observe them quickly. You can observe them under very precarious in vivo situations that we may experience. The relaxation times are so short because of the paramagnetic environment that these protons are in, create a very sensitive scenario. But most importantly, that the chemical shift of these signals, not the amplitude of these signals, but how these signals are located and how they shift is the readout that we observe. Again, so this is a molecule, which is the sensor. So if you change the environment of the sensor, the structure of the molecule will change. And as the structure of the molecule changes, what you then read out, that structural change is from the chemical shift. And so this is encapsulated here. This is a molecule that we are using as our sensor. These are the types of signals that we observe. Using this as an example, the H6 proton, it has a pH sensitivity, but obviously any molecule reports environment by change. Is that these molecules have, or molecules like this, have very, very short relaxation times. So you can observe them quickly, and therefore imaging these have been possible. And the readout from them in terms of chemical shift is not confounded by other overlapping signals. So this is an in vitro example of this kind of work from my colleague, Daniel Corman. And this method is called biosensor imaging, redundant deviation shifts, BIRDS for short, essentially showing you that in these two phantoms, which have very different pH, you can read out the pH with high confidence, even if you change the temperature. So you can essentially read these two things out simultaneously. The key thing is that this is not like most imaging contrast. This contrast is not based on the amount of agent that you have in the given compartment, but really their chemical shift. So it's a readout that's independent of concentration. And so using this method, we've been able to apply it to look at the cancer environment, specifically the pH in the extracellular space, in a variety of different scenarios. These are examples from brain tumors specifically, shown for two different types of tumors here, but you can note and appreciate the acidity within the tumor core, as shown here, core being identified by the line that shows up in the MRI contrast. But you can also appreciate this extended area of acidification for this tumor and not that tumor. And that has to do with how evasive this tumor is, and using cellular markers, you can corroborate that finding. So this goes back to the point that our pH readout, so this is a case of a very non-evasive tumor versus this one. These tumors invade, and this invasion, if you will, we believe is essentially preconditioning for the expansion of this tumor within time for these two specific cases, U87 and RG2, and not the 9L. So I've talked about a little bit about what we do with the proton imaging part to read out the pH, and that uses the proton nucleus. All right, this is a very highly sensitive nucleus to observe because of the high gyromagnetic ratio. But what I'm going to talk about next is the sodium imaging part, which has a lower gyromagnetic ratio, but aided by the fact that we are looking at a quadrupilium nucleus, we can benefit a little bit from the shortened relaxation times of this nucleus. So that's kind of where we are. Our basic rationale for this type of experiment is that the problem that we face is that the sodium signal, whether looking at the interstitial or the intracellular pool signals, they overlap. They're not chemically different. And therefore, the total signal you observe is a representative of these two types of signals. Our expectation is that somehow we can separate the two. And that separation will allow us to look at things like the transmembrane gradient. So the idea stemmed from work that existed within the field, but we advanced it in the last few years, is that if we have a polyanionic paramagnetic agent, much like PET agents that I've talked about, and we take a sodium ion, because of its negative charge, it's attracted to it, and there is pseudo-bounding of some of these ions because of the negative target of this agent, and we have some exchange of free sodium with this sort of bound sodium. And if that exchange happens fast enough that we can reflect the shifting of the sodium signal from this process to reflect the two compartments of sodium. So in essence, what we do, and this is our rationale, take before we add an agent like this is the total signal we will observe. And when we inject or introduce an agent like this, because it's so negative and it can attract positively charged sodium ions towards it, we can separate these two sodium compartments. And this is a theory which we've contributed to, and two key factors that we point out in this both theoretical and practical demonstration of agents like this is the fact that there is a certain bound fraction of sodium towards these agents that's important, but also their exchange. And these two mechanisms, F and Kx, actually contribute to how much the sodium signal will be shifted, so the shiftability factor, but also how these signals will be broadened because of their paramagnetic nature. So another key thing that we point out is that these shifted signals, broadened signals of sodium that we will observe is dependent on the sodium concentration, on the agent concentration. And here we are limited by the fact that we need sufficient amount of this agent present to cause this shifting or the broadening. And therefore, this is something that will act as a benefit for sodium sensing, but it was not necessary for the proton sensing. So our expected idea for these experimentation is that before we add the agent, we're seeing one signal, and after we add this agent, we're going to separate the signal. Now you see 3, and I point out 3 because you also have sodium, plenty of sodium, also in the blood compartment, but because the blood compartment is much smaller than the interstitial compartment, these representative amplitudes of these peaks are depicted by. So in vivo, inside a tumor voxel, this is what we see. This is before the agent, it's been separated here. And outside of the tumor, we see something that's similar, but to the shifting and the broadening is to some lesser extent. So this is kind of what we typically do nowadays, and this is work by Mohammed Khan, who did his thesis work on this project, this is the conventional location of the tumor. We inject the agent to identify the blood compartment, the interstitial compartment, the intracellular compartment in terms of sodium. We integrate these signals to get maps like this. And using a combination of these two, we can get what's called an endothelial gradient. This is a sodium gradient representing the endothelial compartment. And this is the critical one that we will talk about, which is the transmembrane gradient, which is obtained from the intracellular and the interstitial signals of sodium. And we can now map this in 3D to get the transmembrane gradient, how it appears within the tumor. I point out there's these blobs of high intense signal represents the ventricles. Ventricles has a lot of sodium in them, and that has a variety of ramifications for other disease disorders as well. But the key thing is that we can obtain a clear readout of the transmembrane gradient shown for 3 different tumor types. And so this is a pretty uniform and ubiquitous observation that we can now make. We've even begun to employ these methods for a variety of different treatments. And so this is a very unique anti-angiogenic treatment. And what is shown here is a comparison of what sorafenib does. It blunts, it sort of impedes the tumor growth, which is most significant within 2 weeks. And this kind of effect can be read out by the pH as well as the transmembrane gradient. As you can see, the sorafenib begins to normalize the pH with treatment, and as well as strengthening the sodium transmembrane gradient upon treatment within the tumor, making it similar to the normal tissue as well. So I hope what I've been able to show within the last few minutes is that we now have a probe which can sense both proton and sodium. A lot of credit to both Geraldus and Sherry, who designed probes of this particular probe and probes like this, which with proton imaging can be a very powerful proton sensor for pH imaging, but also a sodium sensor in terms of its signal shifted. It can be also used simultaneously. So I hope what I've shown is that all cells, not just excitable neurons and muscle, generate and receive bioelectrical signals that are encoded within changes in the transmembrane potential and the ion fluxes that occur at the cell membrane. And these things happen regularly, on timescales of milliseconds, seconds, to even days. But these are inextricably regulated by metabolism. And that is the connection that is needed to find the proper readouts of various treatments that we see. And I think this is another way of saying this, is that we need advanced in vitro imaging methods to assess the non-genetic biophysical aspects of tumor microenvironment that regulates balance between normal growth, but also the disorganization that happens with most solid cancers. But importantly, simultaneous imaging of tumor invasion and proliferation can hold a promise for early cancer diagnosis and tracking therapies. We believe from chemotherapy to immune therapy. We're always grateful for NIH support, and this work is really a collaboration among many colleagues within our group. Mohammed Khan and John Walsh are PhD and MD PhD students who were involved in a lot of this work, but also my colleagues in terms of postdocs who are still present, Dr. Kumar Mishra and Mihailovich, but also my colleagues within radiology and neurosurgery, Dr. Coleman, Zhao, and Paulitano. And I thank you for your attention.
Thanks very much. I think that was great. You mentioned chemotherapy and immunotherapy, and our next talk is from a radiation oncologist, and one wonders whether this might also help identify tumors and track tumors that are being irradiated, since as he well knows, that is Dr. Hansen, it is sometimes confusing after someone's been treated whether there's active tumor or not. So without further ado, James Hansen is an associate professor of therapeutic radiology and chief of the Gamma Knife program in therapeutic radiology. He received his medical degree from UCLA School of Medicine and a master's degree in biochemistry and molecular biology at UCLA as well. Dr. Hansen's clinical area of expertise is gamma knife stereotactic radiosurgery for the treatment of CNS tumors, head and neck tumors, lung cancer, as well as lymphoma, skin, GI, and GU. I will bet that he spends more time on CNS disease than other things, but he can tell us. His research focus is studying the interplay between autoimmunity and malignancy and attempting to harness and optimize select autoantibodies to use against cancer. James, thanks for joining us.
Thank you, and thank you for that introduction. And yes, if you can explain my clinical career to me, that would be great. I would love to understand it myself.
But let's talk.
Yeah. All right, so let's see. Can you see my slides and hear me?
Yeah, it's in presentation. No, that's great. Yeah.
All right, here we go. So I'm going to be talking today about my particular interest in using lupus antibodies against cancer, and in this case, how we can maybe use lupus antibodies against brain tumors. And so Already, I probably have lost half of you that are just saying, well, that's impossible. Antibodies can't do that. But just give me 20 minutes or so. Give me a chance. I think that there's something here that's worth talking about. I do have some disclosures. I'm a consultant. I have grants from inventor in patents licensed by this company called Patris Limited, who has licensed the deoxymab technology. So with that said, let's jump headfirst in. I don't think there's any secret that antibodies have revolutionized our approaches in modern day to treatment of cancer. But I think it's important to recognize that all the antibodies that we really rely on in the clinic right now are targeted towards extracellular targets. So things like surface receptors or circulating growth factors. But doggone it, it seems like there are so many intracellular antigens that if we could just get an antibody in there to engage, we could add an entire new dimension to immunotherapy. But the dogma has always been that that's impossible. Antibodies don't penetrate live cells. Now, this is where critics will often interrupt me and say, that's not true. We have antibodies that are already in clinical use that are internalized. What about Kadcyla, the TDM1s? And I would say, I say the nay, that's not what I'm talking about. I'm talking about an antibody that can get in and engage its actual native antigen. In these ADC constructs, these antibodies are still looking for surface receptors like HER2 in this example. And then the antibody gets eaten up and destroyed in the endosome and lysosome, which is great for this mechanism because that's what we want. We want that drug to be released, but that doesn't utilize the exquisite binding specificity of an antibody against intracellular targets. So I think we need an antibody that can get into cells and not get stuck in those endosomes. But how are we going to do that? Well, I guess we could try to invent one. We could stick things like cell-penetrating peptides onto antibodies, like the TAT peptides and such. And that's been tried, but those tend to still get stuck in the endosomes. I guess we could try gene therapy, but boy, that's got all kinds of challenges. I'm going to leave that to other people. Maybe if we can just find a naturally occurring antibody that penetrates cells and use that as a platform to teach us how to invent these antibodies, that seems the most appealing to me. But anybody have any idea where we're going to find an antibody like that? Well, how about in lupus? So systemic lupus erythematosus. It is the prototype autoimmune disease. Patients suffer widespread tissue destruction and inflammation as their immune systems recognize their own cells and tissues. Now, one of the laboratory hallmarks of lupus is the presence of circulating autoantibodies that are reactive against the patient's own DNA. Now, those antibodies are a big mystery. We still don't know exactly how they contribute to lupus pathophysiology, but remarkably, it is now finally accepted that a small percentage of them actually have the ability to cross through membranes and penetrate into live cell nuclei. Well, hey, so there we have it, right? We have a source of naturally occurring cell-penetrating antibodies. We can use those for all kinds of therapies, right? Well, hold on now, everybody. We're talking about lupus, right? I mean, at last I checked, lupus is still in the textbooks as a disease. And indeed, a lot of these cell-penetrating lupus autoantibodies are just broadly cytotoxic, and there wouldn't be any benefit to giving them to a patient. But thankfully, that's not true for all of them. There's an antibody called 3E10, which is the hero of this story. It was discovered in the early 1990s at UCLA by Richard Weisbart, who's pictured there at the left, along with his technician Grace Chan and his great colleague Robert Nishimura. They're great friends and colleagues. What makes 3E10 so remarkable is that it was isolated from a lupus mouse. It penetrates extremely effectively, specifically into the nucleus of live cells, so it does not go through endosomes, and it does not kill or is not toxic in any way to normal cells. So now we've got our chance. Now we have an opportunity to use a platform antibody that penetrates cells. And in fact, as we dive a little deeper into how this antibody really works, it turns out this antibody is really well situated for targeting things like tumors or sites of tissue damage. And that is because part of its mechanism of penetration is dependent on presence of extracellular DNA or nucleosides in the area. So what we're showing on the left here is something we call the 3E10 bullseye effect. The dark stain represents where the antibody is. And as you see, that dark stain is getting lighter and lighter as you get further out from the center. And that's because there's a dead cell there in the middle. And it's releasing DNA into its surroundings and helping the antibody penetrate the live cells that are closest to it but less effective as we get further out. And in the middle, we show we can reproduce this in the laboratory just by adding DNA to the antibody. At the bottom, we allow the antibody to penetrate 100% of the cells in the culture. So the DNA has to be there. And then if you take this antibody to mice and you give it to mice that don't have any tumors or damage, you don't see the antibody really going anywhere. But if you give it to a mouse with a tumor that is necrotic and is releasing DNA, you do see the antibody localizing to that tumor. That's what's shown in the top right. The brown stain are the nuclei penetrated by this antibody. So what on earth is going on here? Why is this antibody using DNA to penetrate cells? How is it doing that? Well, that's a little bit of a longer story than we have time for today, but just to jump to the punchline, it is using a specific nucleoside salvage pathway to get into live cells that are salvaging DNA and nucleosides from their surroundings. And that transporter is called ENT2. It is my best friend transporter in the whole world. It stands for Equilibrative Nucleoside Transporter 2. So in order for 3E10 to cross membranes, to penetrate into cells and nuclei, you have to have two things. The cell has to express ENT2, and there has to be DNA or nucleosides around that cell so that the antibody can bind to the nucleosides and then follow them through ENT2 into the cell. And that's why the antibody likes to preferentially accumulate in vivo in sites of damage, like tumors, where DNA is being released. And ENT2 is salvaging the DNA, and along with it, the 3E10. And that works for injury sites as well. And that's what we've seen in multiple studies. For example, moving from left to right, if you take the 3E10 antibody and you link it to a heat shock protein, and then give it to mice or rats actually that have had strokes, you find the antibody gets the heat shock protein into the ischemic brain and improves neurologic function and recovery. In the middle here, we're showing heart attacks in rabbits. The 3E10 antibody finds the site of the heart attack and delivers the heat shock protein. It works remarkably well. And on the right, we're looking at tumors. You take the antibody and you fuse it to P53, that protein from a long time ago, and it absolutely localizes to tumors and shuts them down. So 3E10 has great potential as a delivery vehicle for tumors or sites of damage. And there's now more companies looking at this than I can even count. But that's not all the antibody does. It turns out that it does more. It's not just a delivery agent. When the antibody penetrates into the nucleus of a cell, it will bind DNA. But if it has its choice, it will preferentially bind to DNA that's broken. And then it's going to mess around with DNA repair. So it blocks base excision repair and Rad51 mediated homologous recombination. And that means it can make cancer cells more sensitive to DNA damaging therapy, like certain chemotherapies and radiation. But even more significantly, if the cancer cell or the tumor has a pre-existing defect in DNA repair due to a mutation such as BRCA or PTEN loss, the antibody doesn't need radiation. It doesn't need chemotherapy. It will kill that cancer cell by itself by causing persistence of DNA double-strand breaks. But that doesn't happen in normal cells that have intact DNA repair. So we have a selective toxicity for HR-deficient tumor cells with this antibody. So we reported that quite a while ago. And then we asked the question, is it just 3E10? Is this just something magic about 3E10, or is this true for other cell penetrating antibodies? And it turns out, indeed, it's not just 3E10. There are antibodies that penetrate cells and cut DNA and kill the HR deficient cells. So there's a pattern emerging here where lupus antibodies, some lupus anti-DNA antibodies seem to be toxic to HR deficient cancer cells. And so then that led us to start asking some questions about what does this mean for lupus and cancer risk in lupus? And I'm not a rheumatologist, I did not know, but we started reading and looking. And overall, cancer risk is increased in lupus, but it's driven mostly by hematologic malignancy. So if you back up and you say, well, let's go tumor type by tumor type, you get a surprising finding in that breast cancer occurs at a lower than expected rate in lupus. And this has been widely recognized for years, but no one can figure out exactly why. There's no clear associating factor. But if you look a little deeper, it looks like it's the triple negative breast cancer that is specifically suppressed in lupus. And we do know that triple negative breast cancer is associated with the BRCA phenotypes and the HR deficiency. So then we start to think, well, if that's true, like why aren't lupus patients making these triple negative breast cancers as much? Well, we know about these DNA damaging autoantibodies that are killing these HR deficient cells. Is it possible that lupus anti-DNA antibodies actually are protective against breast cancer? And if so, maybe we can re-engineer them and use them to treat triple negative breast cancer. And so we're excited about this. We sent it in as an opinion letter. I thought I was extremely clever. I was very impressed with myself when I called it the Lupus Butterfly Effect, because lupus is the symbol of, the butterfly is a symbol of lupus. The butterfly effect is a symbol of chaos. There's chaos in immunology, chaos of using lupus antibodies against cancer. I thought it was a great title. And then the reviewer said, nah, you haven't proven it. You got to call it a theory. So it was published as the Lupus Butterfly Theory. But I will forever want to remember it as the Lupus Butterfly Effect Theory, Hypothesis, Postulate, whatever you want to call it. But we do need to prove it. So we published that in 2016. And we started asking around, anybody can help us with epidemiology, because we have no idea what we're doing in that regard. And it turns out lupus is rare enough that it's a challenge to actually prove the association between lupus antibodies and breast cancer risk. But not an insurmountable one, because Johns Hopkins was able to do it, and they published it just last year. So this kind of blew my mind, and it was very exciting to see this. The Hopkins lupus cohort, they were able to analyze 2,000 plus lupus patients that entered their cohort without a cancer diagnosis, and then evaluate their risk of breast cancer over the years. And they were able to associate that with the patient's laboratory studies and anti-DNA antibody profiles. And we do see finally proof that there is an association between anti-double-stranded DNA antibody positivity and that reduction in breast cancer risk. Patients that make those anti-DNA antibodies had a 45% reduction in the breast cancer risk. And if you dig even deeper and you stratify the patients based on the amount of those antibodies they are making, the low producers did not have any reduction in breast cancer risk, but the high producers had a 59% reduction in risk. So I'm taking this as backing up my theory. I think the lupus butterfly theory can be called the lupus butterfly effect now. So I might ask for a revision to that article, although it's been 5 years here. But regardless, it seems that we're learning something about lupus antibodies and at least breast cancer. And it gives me more confidence in what we're doing in trying to re-engineer lupus antibodies to treat triple negative breast cancer and other tumors. So what are we doing in that regard? Well, again, I remember and I recognize 3E10, although it's technically safe in normal cells, it's still a lupus anti-DNA antibody. And you don't want to give anybody lupus-like symptoms. So we got to rethink this a little bit before we start taking this to clinical trials. The last thing you want is a lupus FC region being administered to patients, because the FC is going to activate complement and ADCC and all kinds of badness. But the good news is 3E10 does not care whether or not it has the FC tail. It'll penetrate cells with or without the FC. It'll bind DNA and inhibit DNA repair with or without the FC. So first thing we've done, cut it off. There is no more FC tail. The danger is gone. And that's how we make what we call a single chain variable fragment, SCFE. It's only the variable sequences of the light and heavy chains. And that's been optimized to increase the affinity for DNA. And then we stuck a couple of those together to make a di single chain fragment to bump up the avidity for DNA. And that works really well against HR deficient cancer cells and tumors. And that's the product that was finally licensed by a biotech company, Patris, as we talked about, who then has helped with funds to allow us to humanize and de-immunize and further optimize the CDRs to develop the antibody I would like to now introduce, named Deoximab-1 or DX1. Deoximab because DNA, mab for the antibody. Yeah, you get it. DX1 is, well, if ENT2 is my favorite transporter, DX1 is probably my favorite antibody, although there are some other ones that are kind of catching my attention too. It works great, penetrates so clearly into the nucleus. We see triple negative breast cancer cells on the left and breast cancer brain met cells on the right. It's killing the cells, it's sensitizing to radiation, but it's still leaving normal cells alone, just what we want. And we are excited to move this towards clinical trial testing. But wait a second, what am I saying something about brain met cells? Am I arguing that we could maybe be using this to treat brain mets? I mean, this is an antibody, right? I mean, the story is already kind of far-fetched, but brain mets, well, the thing is, one of the key biologic markers that predict sensitivity to our antibody is loss of PTEN, even more so than BRCA. And I know that all the DNA repair experts are probably jumping up and down right now saying, well, the PTEN HR link is not completely proven, and fine, but cells that are PTEN deficient are killed by this antibody. And brain mets often exhibit PTEN loss, even when the primary tumor is PTEN positive. And that's either evolution towards metastasis or secretion of PTEN suppressive microRNAs by astrocytes. It's also worth noting PTEN is lost an awful lot in primary GBM. So maybe we've got treatment for GBM as well. But I hear what you're saying. Brain mets, brain tumors with an antibody. I don't understand why the skepticism. I mean, all we need is DNA and nucleosides and the ENT2 transporter, right? It seems like the antibody should be able to get into the brain tumors just fine. All right. The blood-brain barrier. I guess that does pose something of an issue. Maybe I should have thought this through before I signed up for this talk. I guess it's too late to back out now, though. If we look really closely at the blood-brain barrier, and we look in at the brain endothelial cells, whoa, there's my buddy, ENT2, the luminal surface of the brain endothelial cell, and it actually regulates nucleoside flux into and out of the central nervous system. So wait a second, this is all starting to come full circle now. I think I'm arguing that our antibody DX1 can get over the river, the bloodstream, and through the woods, the blood-brain barrier, and into the brain tumor we will go, where the antibody will then bind to DNA and inhibit DNA repair and kill those brain mets, and also the possibly glioblastoma. And we are seeing that this indeed is working out very well. Let's see, is there somebody? Sorry. We are finding that DX1 does cross transwell models of the blood-brain barrier, and it does so in an ENT2 dependent manner. If you shut down ENT2, the antibody can't get across. In the middle panel here, we're showing mice with orthotopic brain tumors, GBM in this case. And we gave the antibodies, we gave the mice antibodies labeled so that we can see it on imaging. And the antibody gets into the brain tumor, great, but not if we treat the mice with an inhibitor of ENT2. So you shut down ENT2, the antibody can't get into the brain tumor. So it's working like we think. And it seems to actually matter. It makes a difference. The panel on the right, these are mice treated with the antibody with PTEN deficient patient-derived GBM. DX1 by itself, no radiation, no chemotherapy, significantly suppresses the tumor growth and extends survival. There we go. Brain mets are a little harder to study. GBM in mice, just implant the tumor intracranially and watch, and it goes. But that's not really fair for a brain met model. So the way we study brain mets has been taught to us by our good friend and colleague Jiangbing Zhao, and mastered by research associate in my lab, Benedette Kafari. These are not easy experiments. Breast cancer cells are injected into the hearts of the mice to allow them exposure to the circulation. And we use a brain-seeking subtype of the cancer cells to go to the brains. And you can then track those based on their signal on serial imaging. And so if we give the mice brain mets and we treat them with tail vein injections with control or the antibody, just the antibody, no radiation, no chemotherapy, I think that picture up in the top right says it all. It suppresses the brain mets quite phenomenally. And it does also extend survival. So this is looking really, really good. But now I start to hear the words again in the back of my head. I don't know if anybody else can hear them. Do we really need a new way to treat breast cancer brain mets? I think, isn't that what the Gamma knife is for? It's easy, right? You see a brain met, you just send the patient to the Gamma knife. And I would say, oh, no, it's not so easy. First of all, not everybody is a candidate for the Gamma knife. And Gamma knife does work really quite well, but there are risks. Radiation necrosis, as Dr. Chang will attest to, is a problem, and there's a lot of it that occurs. And when disease comes back after the Gamma knife, it's even harder to take care of. And then there are the patients for which Gamma knife is unfortunately not an option, that require whole brain radiation. And even with our fancy spinning of the beams, hippocampal sparing and memanting, it carries risks of neurotoxicity. So I don't think there's any question if there was a way to reduce the need for radiation, or at least lower the dose of radiation required, that would make a big benefit for patients with breast cancer and brain metastases and other tumors as well. So we have already started looking into this, and we have a DoD grant to help us conduct this work. I think DX1 is perfectly poised to lower the needed dose of radiation. And I think radiation is perfectly poised to help DX1 get into the brain mets. Because remember, DX1 is looking for and using DNA to get into the tumors. So a dose of radiation to increase tumor death and release DNA should recruit more DX1 to the brain mets. And the bar graphs are shown on the bottom there. So that's exactly what we see. We see the most DX1 get in the brain mets in the mice that get treated concurrently with the radiation. And take it to the obvious extreme, those mice that get DX1 with the radiation also have the best response. We see the less number of metastases. So it's all working the way we expect. I understand it's all preclinical, it's in mice, but sometimes things start to come together, and it's looking very promising to me. Obviously very biased, and very grateful to Benedette and Marta and Caroline and Anu for all their work with this. It's been tough, but we've hung through here, and it's going really quite well now. And again, can't do any of this without help from your friends. I had to cross out the word little and say lot, because Jiangbing Zhao from neurosurgery has been a great friend and colleague to me for years now. He is an expert in all things related to the brain and brain tumors and ways to treat tumors in the brain, and he's helped us figure out these models. He's very interested in the DX1 antibody. We work together all the time, but he also does some other things, believe it or not, a lot of other things. And just recently had a great paper in Nature Cell Biology where he reported on a new effect of an LRRC31 protein that significantly sensitizes breast cancer brain metastases to radiation therapy. So it's almost like the universe is telling us something, because that LRRC31 protein is great, but it can't cross the blood-brain barrier. I think I just spent the last 20 minutes talking about an antibody that can carry things across the blood-brain barrier. So we got the Hansen lab and the Zhao lab working together, and we can put them together and make a DX1 LRRC31 fusion protein that I'm hopeful is going to be next in line to cross over the river, through the woods, and into the brain and increase sensitivity to radiation therapy. So hopefully that was of some interest to people listening here. I have a lot of people to thank. Obviously, Jiangbing Zhao, great colleague. Joe Contessa and Marta Barrow in his lab have been incredibly helpful to us. My own lab, I'm extremely grateful to your efforts. I'm usually at the Gamma Knife nowadays, so you probably are wondering where I've been. Here I am, if you're looking for me. It still looks like me, right? And the Yale Gamma Knife team. Everybody is great help. I'm appreciative to everybody. But last, anyone that knows me knows that I'm a fan of the Marvel movies. And the best part about the Marvel movies is always there's one more cutscene at the very end, right? And I think if you listen very carefully and you look off into the horizon, you will see that there is a team becoming assembled, led by PI Megan King, getting the best breast cancer brain researchers together to expand and improve DNA-targeted therapies towards better breast cancer treatment, with Pat LaRusso, Megan King, Abhi Patel, Ryan Jensen, myself, and Jiangbing Zhao. And we're very thrilled to have been notified just recently that we've received the YCC Team Challenge Award to conduct this work. With that, I will take a breath and stop talking and answer any questions if there are.
Well, I think we're a little short on time, so it's exactly 1 now. James, that was great. I'm certainly extraordinarily supportive of anyone who wants to study breast cancer brain metastases, an area that I've thought about a lot over the years, and I would agree with you 100% that it's an area that's perhaps the most in need, and potentially the most in need in breast cancer, because we may well be able to eradicate disease elsewhere in virtually everyone in the next decade, but the brain is the hardest place, it seems. So with that, I want to thank both of you for great talks. It's been a great grand rounds, and James, congratulations on the challenge award.
Thank you so much.
All right. Fahmeed, thanks again. Bye-bye. And thanks to our audience.